Hello there. Welcome to the world of sustainability where your academic needs are met. Today, we'll be looking at the topic nitrogen metabolism of the liver or by the liver. So today, we'll look one kind of topic which they call nitrogen metabolism by the liver. You understand? So what is the point? What is the point of this topic? First things first, when the body is starved, under starvation states, when the body is fasting, usually during prolonged fasting, the body switches to, normally the body depends on glucose as a source of energy. But under prolonged fasting, when the glucose stores are depleted, the body switches to an alternative source of energy, which is amino acids, or which are amino acids. That's principally gotten from the glutamate proteins. So they are used as alternative sources to generate energy. So during this process of energy generation by these sources of energy, which are amino acids, first thing that happens during the process is usually deamination, whereby the amino group is removed and this carboxylating is gotten. Carboxylating is what is used to generate energy. The byproduct of this process generates amino groups, or call it an it's actually principally called ammonia. So ammonia is therefore generated as a byproduct. Now, this byproduct ammonia, it's not ideal for the body, it's not good for the body. So the body is susceptible to the effect of this ammonia. Ammonia at elevated levels affects the physiological functions, such as the brain, the mental function. It affects the brain, it affects the organs of the body, like the liver and so on and so forth, the kidneys and so on and so forth. So generally, ammonia is harmful to the body at high levels. So the body has a mechanism to actually curb this situation or reduce the effect of this excess ammonia. So that's what we'll be discussing today. So excess ammonia can be taken up or carried by two important molecules, either alanine or glutamine. You understand? So as to prevent the effects of the elevated levels in different parts of the body, like the brain and so on and so forth, and the other internal organs, essentially internal organs. So the body takes this, that's the alanine, and glutamine takes this amino acid, that's ammonia, takes them, the immune group takes them to the liver and deposits them in the liver. And the cells of the liver are called hepatocytes. The hepatocytes, they contain, obviously, every cell must contain or must have compartments like the cytosol and the mitochondria. The principal sites here are usually the mitochondria and the cytosol, as we'll look at shortly. So the liver therefore combines this amine with ammonia with the CO2 to form Carbamoyl phosphate, and which in turn enters the Urea cycle, which is also an important part of an important pathway in the mitochondria of the cell, and converts it to, and is converted through the ornithine to citrulline. So now we'll be looking at the pathway proper. The pathway is talking about the cell, the liver cell, which are the mitochondria and the cytosol. After the carriers, which are alanine and glutamate, or glutamine, have brought the excess ammonia to these hepatocytes. They are deposited. The ammonia molecules are deposited and utilized to form an alternative form, which is glutamine. So glutamine is used to carry the ammonia and taken to the mitochondria, which is therefore converted back to glutamine to release the ammonia that has been taken up already. So this released ammonia is used by the body to form carbamoyl phosphate. So this part might not really do justice to the explanation, because there are two enzymes here that helps to make this conversion from glutamine to glutamate and glutamate to glutamine happen. The first one, which is here, this is the glutamine synthetase. That guy is usually found in cytosol, while the other guy, which is glutamine, is found in the mitochondria. Understand? So the goal is to help carry ammonia safely to the different compartments of the hepatocytes. The process now requires a lot of energy to happen. You understand? So energy in form of ATP. So the ATP, one of the reasons why this happens is that ATP is utilized to convert this these guys, you understand, which are the glutamate and glutamine. So this forms carbamoyl phosphate, and the enzyme there, which is the rate limiting enzyme of the pathway, which is urea cycle, is carbamoyl phosphate synthetase 1. Is carbamoyl phosphate synthetase 1. So this guy serve as a point of regulation of the pathway. So take note of that. And this carbamoyl phosphate converts to form citrulline, and citrulline then use the mitochondria to the cytosol to convert to form adenosine. Please take note. And the enzymes that are crucial in this aspect, they are the ornithine transcarbamoylase to form citrulline. That happens when carbamoyl phosphate is condensed with ornithine to form citrulline. Then the conversion of citrulline to adenosine is aided by adenosine synthase. And there must be another amino acid that would be used in the process, which is called aspartate. You understand? Yes, take note. And the process requires another enzyme again to convert the adenosine to adenine, which is called adenosine adeninosinase. So please take note of that. And this forms a byproduct, which is fumarate. Fumarate can be used shortly into the CTC cycle to be used as an intermediate in the CTC cycle to generate energy in the process. So it's an important byproduct of that reaction. So the adenine is therefore taken back. So all these back and forth movements are aided by shuttle systems to carry them between the two compartments of the cell. Between the two compartments of the cell. So very, very important. So this adenine is used to form urea. And urea is formed by the action of adenase. Adenase forms urea, which is the main product, and also forms back ornithine. Then the cycle goes on and on. And that's why it's called urea cycle, because it has a vicious cycle, goes on and on. And that urea is taken away from the mitochondria into the cytosol, which then leaves the hepatocytes and gets transported to the kidneys for proper excretion. You understand? So therefore, converting the toxic form, which is ammonia, to a less toxic form, which is urea. So take note of what I've just said. Very, very important. You understand? So I try to talk about and say, since the way they carry the ammonia, where they cause problem for the body now, alanine and glutamine. These guys now, they all carry upon the cytosol of the hepatocyte, the liver cells. Then the enzyme really crucial for this process now, glutamine, synthetase, and glutaminase. So this pathway, therefore, might not really do justice to the different compartments or different constituents of the metabolic process. But my explanation will actually help out to elucidate what is actually happening, or what would tell you what happen. So please take note. This whole process, therefore, goes to form the carbamoyl phosphate, citrulline, all these things they happen in the aid of enzymes. And these enzymes are found in the structural part of the cell. I remind you, if I round off, take note that you see the structure of urea has two amino groups. Please take note, they may ask you which part of the pathway are the amino groups coming from. Please, the first one is coming from that conversion of glutamine to glutamate, which releases one, and the other one is coming from aspartate. Please take note. You understand? So from the structure of urea, you look up the structure of urea, you realize what I've just said. It's very, very important. So at this juncture, I would try to give you certain questions to ponder over. You understand? This question will bring you up to speed as to what I've just said or what I've just explained, and it will help you, guide you as to it will give you a better understanding of the concept. Please take notes. If you have any questions, any concerns, you can put them on the comment section. Don't forget to like and subscribe and get more exciting and interesting videos from me. Don't forget to maybe just really push this ministry on. You understand? Forward. You understand? So I'm gonna just follow up. If you have any issue, you're gonna reach out. You understand? So thank you very much. I remain sustained for your sustenance, and you provide academic needs and necessary everything we need with the giver. You understand? So please, thank you. God bless you.